Hello and welcome to Life of Sciences. In this video, we are going to study the topic acquired immunity. So let us start the topic acquired immunity. Students, as we know that acquired immunity is the immunity that we acquire or adapt by infections and vaccinations. Now let us study various features of acquired immunity, and these are specificity, diversity, discrimination between self and non-self cells and molecules, and memorization. Now let us study all these features one by one. So very first feature is specificity. As the name indicates, acquired immunity is specific for specific microorganisms. For example, if a person is infected with smallpox, so he got immunity for smallpox only, not for chickenpox, measles or any other infections. Students, next feature of acquired immunity is diversity. So acquired immunity can recognize a vast variety of foreign molecules and antigens. So students, next feature is to discriminate between self and non-self cells and molecules. So acquired immunity can recognize and respond to foreign molecules or cells and tries to reject them, while for self cells and molecules, this immunity does not show response. If acquired immunity stops discriminating self and non-self cells and molecules, then a condition called autoimmune disorders resulted. Rheumatoid arthritis, diabetes type 1 are some examples of autoimmune disorders. So, next feature of acquired immunity is memorization. Acquired immunity has a strong memory as it keeps the memory of each and every encounter. The first encounter, which is known as primary immune response, is slow and feeble, while the secondary response and subsequent encounter with same pathogen is rapid and heightened, which is known as anamnestic response or booster response. So, primary response takes long time to recognize and respond to antigens, as during primary immune response, after an initial contact with an antigen, no antibodies are present for a period of several days. Then a slow rise in the antigen titer occurs. First immunoglobulin M and then immunoglobulin G, followed by gradual decline in antibody titer resulted. Thus, a person gets sick in primary immune response, while secondary immune response is memory based. Now students, let us study components of acquired immunity. So there are two components of acquired immunity: humoral immunity or antibody mediated immunity, and cellular immunity or cell mediated immunity. Now let us study each component one by one. So very first is antibody mediated immunity. This immunity is provided by B lymphocytes or B cells. B cells are produced in bone marrow by the process called hematopoiesis. These B cells mature in bone marrow as well. As B cells produce antibodies, thus this type of immunity is also known as antibody-mediated immunity. These antibodies circulate in humors or body fluids like blood plasma and lymph, thus also known as humoral immunity. This immunity protects the body from viruses, some bacteria, and toxins produced by pathogens by various ways, like agglutination, opsonization, precipitation, neutralization, lysis. Now, next is cell mediated immunity. It is provided by T lymphocytes or T cells, hence also named T cell immunity. T cells are produced in bone marrow by the process hematopoiesis, but these cells migrate via body fluids to thymus and mature there. So, the next is types of T cells. So students, there are different types of T cells: helper T cells, cytotoxic T cells, memory T cells, suppressor T cells. So if we talk about helper T cells, as the name indicates, help B cells to generate antibodies, while cytotoxic T cells are capable to kill microorganisms and even some of the body's own cells, thus called killer cells. Memory T cells can recognize invading antigens even after several years of first encounter and remain in lymphatic tissues like spleen, lymph nodes, etc. Suppressor cells are capable of suppressing the functions of cytotoxic and helper T cells. They also inhibit the immune system from attacking the body's own cells. Now students, let us discuss various types of B cells. So B cells may be plasma B cells and memory B cells. Plasma B cells can live for only a few days, but they secrete large amount of antibodies by activated B cells, while memory B cells as compared to plasma B cells have longer lifespan. These cells remain dormant until activated once again by a new quantity of the same antigen. So students, let us compare cell-mediated immunity and antibody-mediated immunity. So first of all, let us see cell-mediated immunity. It is due to T lymphocytes which produce different types of T cells. It defends the body against all pathogens. It reacts against transplant, that is, this immunity is responsible for graft rejection. Thus, the patient that has undergone organ transplantation has to take immunosuppressive drugs for whole life. Example of immunosuppressive drug is cyclosporin A. CMI also provide immunity against cancer, while antibody-mediated immunity is due to B lymphocytes which produce antibodies by the help of T helper cells. It defends the body against viruses and bacteria. It does not show response to transplant. It does not provide immunity against cancer. Now students, let us discuss types of acquired immunity. So acquired immunity may be active and passive. If we talk about active immunity, in this immunity, person's own cells produce antibodies in response to infection or vaccination. It is slow and takes time in the formation of antibodies, but is long-lasting, durable, and effective. Active immunity may be natural or artificial. Natural active immunity. So here a person who has recovered from an attack of measles or mumps develop natural active immunity. So this immunity is due to infection of a pathogen, while artificial active immunity is the resistance induced by vaccines like BCG, OPV, where live, weak, genetically modified, killed pathogens or toxins of pathogens are introduced into the body to provide primary immune response. So it is based on memorization of immune system. If we talk about passive immunity, in this immunity, ready-made antibodies are provided. These antibodies are produced in other organisms, say hot serum, and injected to a person for immediate relief. It is short-lived, less effective as compared to active immunity. Passive immunity may be natural or artificial. If we talk about natural passive immunity, here ready-made antibodies, say immunoglobulin G, are provided by mother to her fetus through placenta, while during breastfeeding, initial stages of milk called colostrum is rich in immunoglobulin A. Now, artificial passive immunity is because of ready-made antibodies that are administered as in case of snake bites or anti-tetanus toxin against tetanus. So, this is all about acquired immunity from my side. So students, in next video, I will discuss about structure and function of antibodies. Thank you. Like, share and subscribe my channel Life of Sciences. Thank you.